In this video, I'm going to demonstrate you how to extract metadata from file names in Cell Profiler. When we work with multi-channel data, and this is the case, we have three channels, C0, C1, and C1 simple segmentation. This is a mask image for channel C1. We have to assign name to each input image. So if we have three channels, then we have to assign three names that Cell Profiler will use to do its job inside Cell Profiler. And you can do this naming for all images if you have, let's say, single channel data. But if you have multi-channel, which is the case, you have to use this second option. And then the selection criteria for each channel can be done based on the file name, for example, or extension, directory, a whole bunch of options. So we're going to cover metadata because it's the most flexible and powerful one. So I'm going to show you how this works, and then we're going to go to metadata extraction and show you how these metadata fields or variables, so to speak, are extracted. So here we are using the metadata, and we're saying if the channel does have matching C0, and then we added the second rule to it, and you can add as many rules as you need just by pressing this plus button. So I don't need the third one, I just need only two. And so we're saying if the channel is C0 and the seg field, short for segmentation, does not have simple segmentation in it, then that image or that input file is DAPI. And then similarly for the other channel, if the channel is C1 and seg field does not have simple segmentation, then that's the image called MIDO. Here is the rule for that mask file or mask image, and here we're saying that if the channel is C1 and the segmentation field actually does have simple segmentation, then call it MITO_seg. And these metadata values are extracted in this module, metadata. So of course we selected yes, by default it's no. So you select extract metadata, and then you based on how many different patterns you have in the input files, you need that many extraction methods. Here we're gonna write one extraction method for both channel C0 and C1 because pattern is similar. The only thing that differs is the channel number. However, the pattern for the mask file or segmentation result is different. That's why we have another extraction method for the file names that have that simple segmentation added to them. And you can add, as many extraction methods as you need. And the how this works, if you press this magnifying glass button, you're going to open regular expression editor, and in this editor you just need to find you need to write a kind of template or formula in this field that would extract substrings from the file name and assign them to these color coded variables. As you can see, this project variable has this substring taken from the file name, and then the image name is highlighted in green. Image number is number 1 right here, and then we have a couple more. So this formula for regular expression is basically something within the parentheses and a separator between them. So the expression within the parentheses defines the name of the variable, and you have to write it in this form. In this this is the syntax. And then dot star means all of the symbols up to this divider. So we are using this whole expression as a divider between the fields or the variables, and it could be a different symbol. It could be just one symbol. For example, here we are using let's, for example, a shorter expression, and here we're using this minus sign with two spaces on both sides as a separator. So one advice, if you write, when you acquire your images, if you write them with underscore as separator of the fields, that would be easy to write this. Then you would have only the same underscore as a separator between fields or between variables. But it's not necessary. So you just need to understand how this works. Basically, variable and the length of the variable in terms of symbols, and a separator. So variable, separator. Sometimes the file names can be long, and there may be information that that's not really needed, and I am calling that here, for example, as bogus, because the bogus field that has this value, I don't really need it. That's why I just called it bogus. So if we go from the beginning, so this sign here tells Cell Profiler to start from the beginning of the file name, and then our first variable is project, and as I said, dot star is all the symbols until this separator, and that separator is right here. So the separators are ignored. So if something that you want to ignore, you can use that as a separator. And then the next variable is image name, the green, and so on and so forth. And as I said, here I'm using project maximum because it's kind of generic word, because maximum intensity projection operation was done on all images, and since it's generic, it's not useful in terms of identifying with the output. So that's why I am deliberately ignoring this. And you could ask that this XY positions, which actually I really don't need, why I'm not ignoring them. And I could have written them as separators, like one long separator if I want to ignore. However, I am assigning this substring a variable called bogus, and the reason is that because this XY positions of the images are different, because the data was acquired as multi-position acquisition. So that's why, even though I really don't need this information, they are different. That's why I am extracting it as variable, but I just won't use it. The only other useful information here is the channel number. So this is the kind of the formula to extract the necessary information from the file name. And then the second one is, as I mentioned, for the file name that ends in simple segmentation. That's why this expression is exactly the same as the first one. It just has this added variable, one more field in this regular expression template. it's not matching the file name because this file name is simply the first file in the list of the input files. The cell profiler doesn't go to the second one, but we know that the second file name has only this simple segmentation added to it. So this would be our file name that this formula would work for. And as you can see, this simple segmentation word has been assigned to the variable called seg, short for segmentation. And you already saw how I used those variables to assign the names in this module. I want to show you one more example. And here I am extracting metadata not only from file names but from a folder name as well. As you can see, you have two options here. You can extract it from folder names, and this was the case because the acquisition was on the Leica instrument, and the Leica instrument has this option of performing this mark and find experiment, where you mark multiple locations and it automatically acquires the image from those multiple locations. But when you export the data, let me show it as a detailed list. The file names are generic mark and find. For example, for this condition, we have these file names, and for the other one, the names are exactly the same. So we need to somehow differentiate them, and the difference is in these folder names. So those are the unique identifiers in this case. That's why I am extracting the information from the folder name. So that's why I have 1, 2, and 3 extraction methods to get the useful information from these input files. These two, as I showed in the previous the example, for the file names, for the original file names and the file names with the segmentation result, which has one extra field, and this is just a formula or template to extract the information from the folder names. Again, the variable is within the parentheses. We could have used this, so it doesn't matter in this case. However, you can start this extraction from different portion, but here we're saying, okay, go all the way to the beginning of the string or file name, and I'm using this expression as a separator between two variables, and this is also kind of the information that I am not using or not extracting. So only the path to, as you can see, this is the folder name that is different between different samples. That's how I'm extracting that, and the rest is the same. So, and why this is powerful, why are we extracting this metadata? One is, of course, we're using them to assign names, but there is also another place where they come in handy, and that is to add those metadata to the output information or output data. For example, this pipeline saves the output images using the names of these variables. Here I'm using that path2 underscore image name as the base name for the output image file, and how you want to do it or how you can do it is simply in the file name field, you right click of the mouse, and then you have a list of all of the metadata variables. So from here I choose path 2, and then I'm typing underscore as a separator, and then right click again, and from the list I'm selecting image name. So instead of having, let's say, generic name analyzed images numbered 1 through whatever, this is more informative to have as the names for the output images. Also, you can use those metadata in the Excel files as well. For example, here in the export to spreadsheet module, you have an option of adding the metadata columns to your object data file. So each of the fields of each of the metadata will be added as a column to the Excel file, so that you can easily identify what measurement belongs to what image or what treatment, and so on and so forth. And the object data files that we're exporting are listed here. We're exporting cells with DAPI, the endosomes identified as puncta. So several Excel files will be exported from this pipeline, and all of those metadata will be added to these Excel files as new columns. If you want to add them to the measurements that are done as per image, and then, well, in this case we're not exporting image, but if we were exporting image Excel file as well, then the metadata is available here to select and add into the image Excel file. For the object data files, the selection is not possible. It will just add all of the metadata as columns into the Excel file. But then the ones that you don't need, you can simply remove from the Excel file. So that is very useful tip to extract and use the metadata from file names in Cell Profiler.